Hi, my name is Dan Barton. I'm presenting to you some work on seabirds. I'm probably the least marine biologist person in the room. I'm an ornithologist by training, but I will try to relate the results that we have as best I can to work that, like what Eric was talking about earlier, how seabirds might be an indicator of changes in oceanographic conditions. We do have some evidence of that in the results that we found, and also how seabirds may sort of directly benefit from marine conservation efforts and that they're a part of marine ecosystems in general. This is the work of a ton of people. Yeah, that's not going to do it, so we'll do this. Co-PIs here, really, Rick Golightly got me involved in this, so if my talk isn't very good, you can blame him, because he invited me into working on this project in this room, sort of. Dan Robinette and Hamiyaki from Point Blue, Rec Tyler, University of California, Santa Cruz, several other folks are in the room, Eric Nelson, from U.S. Fish and Wildlife Service, Jerry McChesney as a collaborator provided some data, Ken Griggs has provided a bunch of different kinds of support. I have to acknowledge the loss of a really important figure in the history of monitoring and research and conservation of California seabirds, who was a contributor to this project. Terry Carter just passed away recently. He was a part of this and many, many other projects. A lot of indispensable staff and students: Phil Capillo at UC Santa Cruz, Steph Schneider, who's going to be giving the next talk, Shannon Murphy, who did collect a lot of these data, a bunch of other HSU grad students, and sort of collaborators from Point Blue, and then some partners who provided a lot of logistical support, including the BLM, California State Parks, and Talawa, I just want to make sure I do it right, Talawa Dini or Dina? Dani, thank you very much. Jay Tuck and Rosa worked with us to get us access to some of our northernmost study sites here. And of course, funding and support from California Ocean Science Trust. So rationale for seabird monitoring, I need to convince you these things are important. First of all, they're uniquely observable apex predators. So compared to a lot of other apex predators in marine ecosystems, they're accessible to monitoring because they breathe air and nest on land. They're unique functional groups in marine ecosystems, so they often play critical roles in marine ecosystem processes. They may benefit directly or indirectly from marine protected areas, which is important as they are critical components of marine ecosystems. And they also may allow us to indicate change caused by MPAs, although sometimes it's not clear what kind of change they are actually indicating. But there is that prospect as well. So real briefly, North Coast Seabirds 101. 4 seabird species comprise the vast majority of seabirds in this region. And those are Brandt's cormorant, western gull, common murre, and Cassin's auklet. The ones bold and underlined are ones that were effectively monitored by this project. There are other breeding species as well, some of which were also effectively monitored by this project. And those were mainly pelagic cormorant, double-crested cormorant, pigeon guillemot, and black oyster catcher, which is sort of an odd one. You might say it's a shorebird. I sort of agree. And there are very large numbers of seabirds, including non-breeding seabird species like these seawaters in our waters, but we're focused here on breeding seabirds. These birds use a variety of nesting substrates. Again, here in bold and underline are the species that we focused on. Notice that all the subterranean burrow nesters are going to be ineffectively monitored by most of the methods we used, whereas most of the surface nesters are effectively monitored by the methods that we used. So diet and foraging locations drove some of our choice of focal species. It also drove some of the methods that we could use to monitor these species. So it's a combination of convenience and biological rationale here. So offshore birds that we effectively monitor: Brandt's cormorants, they tend to forage on fish offshore. Common murre, pigeon guillemot kind of nest inshore or offshore. And then nearshore piscivores include pelagic cormorant and double-crested cormorant. And then there are several species of offshore sort of planktivores here that were not effectively monitored by our project, but are important parts of our seabird community in the region. And then you'll notice that western gull isn't up here because they kind of do whatever they want. And forage in a bunch of different locations, many of which are not really marine areas like a dump, right, or your lawn. So they're kind of an oddball here in terms of their foraging ecology. So our study sites span the entire region. We really have 3 different methods, sets of methods associated with 3 different projects. I'm going to talk about 2 of them. Steph Schneider is going to talk about the other one next. So we monitored seabird colonies, shown here in yellow, from the California border to Point Arena, using aerial surveys. So that allowed us to estimate trends in seabird abundance via aerial colony counts going further back in time than the MPA process, back to 1989, thanks to the existence of historic data here. We conducted shore-based monitoring in 2014 and 15, and then a small citizen science cross-validation, potentially to evaluate the effectiveness of citizen science for monitoring seabirds in the future. And then this intensive productivity and diet monitoring study at Castle Rock done via remote camera system that the U.S. Fish and Wildlife Service, Eric Nelson, Rick Golightly, other folks, and Steph Schneider is going to be presenting results on next, so I won't talk about that one. Methods for collecting data on trends in seabird abundance, flying a Barton Avia P-68 over colonies in this region, taking photographs of colonies. There's a large number of archived photographs, some of which we were able to count using funds from this, some of which had already been counted, and some of which remain uncounted. There's still data sitting there that we haven't been able to process yet. You'll see why. It's a labor-intensive issue, and so a lot of this data were shared by other collaborators. So essentially, you fly this plane over. Here's Mike Parker and Phil Capitello taking some photographs out of the belly port of one of these things, a CDFW plane. I'll just avoid one question you may have in your head. Is using drones cheaper? No, not yet, right? This is, we cover the entire region for about $7,000. When drones can do that, I'll be super impressed, and we'll switch to using those. But there's prospects there. So, and then these photos were counted by Phil Capitello, Crystal Shore, and Steph Schneider. So like audience participation time, we haven't had much audience participation today. So here's a photograph. This actually isn't one of the colonies in our sample. This one's from Southern California. The birds show up real easy in this one. So it's an easy one, right? So how many Murres are in this photograph? I'll give you a hint, right? So first, there's some Brandt cormorants in this photograph too. So the big blobs that are pointy, right, and have a long neck, those are Brandt cormorants, and then these are common Murres here, and then there are some nests too, which are just the round things with a blob sitting on top of them. So take a wag, right, a wild guess. 400, 600, 4,000. Okay, well, the mean of those three guesses is about right. So 1,245. So maybe that's what we should do. We just average everyone's guess. So there are 1,245. So this is really labor intensive. And Phil and Steph and several other people did this with colonies that were over 100. Well, Castle Rock is over 100,000 birds. So there's a lot of effort that went into doing this. So the creation of this data set helps repeat something that was done in '89, largely through the efforts of Harry Carter. So back in '89, there was a statewide survey done. It really went into 1991, but the aerial surveys were done in '89. And so a large portion of California seabirds are in our region, and it's about 41% back then. And so the region-wide baseline, we counted all the photos from all the colonies from 2014. And so common Murre, there's about 350,923. That's how many were in the photographs, right? Birds counted at 19 colonies during June. This isn't all of the common Murres in the region, because some of the birds are not at the colony on the day you do the flight, right? So it's not all of the birds of the region, it's the ones that were present at the colony. So you've got to keep in mind this is colony attendance, not really population size. It's a biased underestimate of total population size. And so 41% of those common Murres were at just Castle Rock. This is a huge seabird colony. It's also a special closure near a marine protected area. Brandt cormorants, so there's about 7,000 birds, about 5,000 nests at 20 colonies, 19 of them with nests. Double-crested cormorant, there's about 2,500 birds, 1,800 nests at 13 different colonies. And then brown pelican, don't read here, but there's approximately 1,000 counted in June at 14 different roost sites throughout the region. And so this spans the entire region here, this survey. So the existence of previous data and these photographs that we went back and counted allow us to do a trend analysis. So for common Murres, we had 173 observations of attendance over this 25-year time period. That's about half of the potential year by site combinations. So it's an incompletely observed time series. We could add more data to this by going back into those photographs, but that's really limited by that arduous task of counting the photographs themselves. For Brandt cormorants, we had 123 observations of attendance at 10 colonies. We also had counts of nests for Brandt cormorants, which is I'm going to show you, at 10 colonies during those 25 years. We used a state space model of growth and observations. You may have been told in error there would be no math. Here's what it looks like. So we used a first order Markovian growth model. That just means growth only depends on population size last time. And we're going to get two parameters out of here that are of interest. We're going to estimate population size, and we're going to estimate the annual growth rate of the population, and that's can be thought of as a percentage change per year. We also included in this model, and like Eric and I are going to talk about this a whole bunch, I hope, real soon, an effect of the Southern Oscillation Index, which is a measure of the El Niño event, and so a negative value of this SOI versus a positive value. It's really just a normalized pressure difference between Darwin, Australia, and Tahiti. So this is a measure of El Niño, and negative values, strong ones represent El Niño, positive values represent La Niña. And then we also assumed, so that we could estimate population size in years that we didn't observe those colonies, that variation among colonies and their growth rates is normally distributed, and I'll show you how that works in a sec. And then also that we have normally distributed observation error, so that we our counts weren't actually the population size, they're just the number of birds at the colony that day. So there is some error in the relationship between those counts and the true population size. So what do we see in these kinds of results from this model? So first of all, the circles are actual count data. This is Cape Vizcaíno, right? So down there kind of west of Leggett. And this is a common Murre time series here going from 89 to 2014. The gray ribbon is the 95% credible interval of the population size estimate, because I used Bayesian inference here. And we can see that the estimate of population size generally tracks the observed count data. And the value of using this kind of approach is that we can also infer the historic population sizes over time at colonies where we had less observations than at Cape Vizcaíno. Cape Vizcaíno is kind of an index site. We had almost all the observations. Because it doesn't really make sense to think that, like, oh, because we didn't count the Murres at Blank Rock this year, you know, maybe there were like 7,000 that year. That doesn't make sense, right? So the pattern, even though it was incompletely observed, probably reflects some kind of pattern of growth. And also the uncertainty, though, in unobserved years gets bigger. So summarizing these data across all of the sites we observed, generally speaking, most Murre colonies in the region are growing. There's two weird ones, right? You just sort of see the spaghetti that doesn't fall in line here on the spaghetti plot. And those are Redding Rock and Goat Island, which are two sites which are hypothesized by other folks to have high rates of disturbance, which may have altered the patterns of population growth there. So common Murre region-wide, population's growing. Probably recovering from historic impacts of egging and colony disturbance, as well as the impacts of gillnet fisheries on birds, all of which are sort of well documented. So the average growth rate over the observed interval was 4% a year. Colony-specific growth rates varied from -7 to +17% per year. So there is some variation among colonies in our estimates of those growth rates. Brandt's cormorants, much less clear. So Brandt's cormorants have actually shown a small decline in estimated population sizes over the observed interval, and they're more highly variable through time, but they're actually more consistent among colonies. Sort of interesting. It's a smaller number of colonies with a smaller number of observations here. But so Brandt's cormorants do sort of show a different pattern. Now both of these species appear to be responsive to the El Niño Southern Oscillation Index here. So mean annual SOI is on the x-axis, and this is just from one site, right? This is an index colony here at Castle Rock. This is population growth rate on the y-axis. So down here, El Niños, negative rates of population growth. Over here, La Niñas, positive rates of population growth. Overall, the average is a positive rate of growth. So really for, there's about a 4% difference in El Niño versus a La Niña event, maybe up to into 6% between those. And then this is a measure here of our certainty of the size of this effect, and I'll just say it's high, right? So that's the posterior probability distribution of the slope of the effective SOI on growth rate. And then El Niño and cormorants, we see a similar pattern, not quite as strong. So there's this positive effect of SOI, meaning a negative effect of El Niño events, and there's a little bit less certainty there. Overall, this trend monitoring approach, we find that our certainty in population size estimates is greater than the variation in growth rates, meaning we're fairly certain we can detect changes in population size over time going forward using this method, including potentially changes caused by the existence of MPAs in proximity to these MPAs, or sorry, into these colonies. So moving on there, I mean, I'm really trying to talk about 2 projects in 15 minutes, so this part's going to be a little short. But we also did this near-shore monitoring shore-based approach. So this involved 6 MPAs and a treatment control study design mapped here on the left. Again, it spanned the entire range of this study region. Shannon, how many miles did you drive? Too many, right? So we got this enterprise rental car and drove to all of these sites about 15 times over a summer. So I guess we got our money's worth. Don't tell Enterprise. At these sites, we did nest monitoring, near-shore foraging observations, which means sitting there and counting foraging birds for 4 hours. Rocky habitat use, so just counting the number of birds in these rocky habitat regions. These are all rocky habitat regions, generally speaking. And then disturbance monitoring. So general descriptive results here. This is at one of our sites, right? This is what some people insist on calling Camel Rock, but charts say is Little River Rock, right? That's like one of the great debates in the Trinidad area. What's the name of that rock? So this is one of our sites, and this site included all of those 4 different techniques. We mapped out nests and monitored nests over time. Generally speaking, we find that there's breeding populations of most of the nearshore species that we were able to monitor in all 6 of the MPAs we studied, except for Pyramid Point. And then there's foraging habitat for most of these species as well. It's kind of a little bit mixed up there, but I'm going to move on. Nest success, lower in 2015, consistent with an effect of oceanographic changes. Disturbance rates. In our region, disturbance rates are lower than in Southern California. Probably not surprising. Sources of disturbance in our region include aircraft, watercraft, and people on foot. Pretty typical. So foraging observations, we have a ton of data here. No short-term effects of MPAs thus far. None to report on. Roosting birds, there's actually more birds roosting outside of MPAs. And that has nothing to do with the fact that they're MPAs. Right. So there's just more rocky habitat that was appropriate for seabird nest sites outside MPAs. At the sites we surveyed. And then we cross-validated citizen science by comparing professional data, so like data Shannon collected. You're professional now, right? Yeah. Data Shannon collected with citizen scientist data. It was hard to get people to participate. We did the best we could. We did some outreach, but it was difficult to get people to participate. But those who did collected data that was consistent across sites, just in terms of the count of birds out there, right? That was the only thing that we really tried to cross-validate here. So future and integration, they are uniquely observable apex predators. So they are easy to count. They're easier to count than fish. That was the mantra of our project, right? Birds are easier to count than fish. Especially population growth, and then you're going to hear a little bit about reproductive success and diet here in a sec. There's an opportunity here to continue to establish historic baseline data from these pre-existing data sources. We've done that as best we could up to this point. You've seen the kind of results we could produce. I'm excited about integrating that with more oceanographic data to maybe fine-tune some of those questions instead of just being like, El Niño's bad for birds. We kind of already knew that. Like spoiler, like there was no spoiler. Opportunity for strong treatment control study designs. There really is here for our shore-based work. That was a lot of work, 15 visits to each of those sites each year. So there's like hundreds and hundreds of hours of observation data here that's spatially referenced and is easily repeatable. And then also BACI designs, which we weren't really able to do given the timing of the onset of these projects versus the establishment of MPAs, but we can do that using this pre-existing data. And then additional contextual data beyond SOI, and I just put HELP in here. I guess that means the Farallon Institute and Eric HELP. But with that, I don't think I left any time for questions, apologies, but I'll see you at lunch.